When we talk about fluids, we are referring to water and everything that's dissolved in the water, hence the term fluid. And the average adult contains about 40 liters of water. There's a higher percentage of water in men's bodies because of the muscle mass versus female bodies because of increased adipose deposition. Now we divide this fluid into two major compartments, that being the fluid inside of the cells, or the intracellular fluid compartment, and that outside of the cells being the extracellular fluid compartment. And notice that most of the fluid in the human body is within the cells itself. But there is constant exchange between the intracellular fluid and the extracellular fluid through a series of pathways. The first of which is the interstitial fluid, which is that tissue fluid just outside of cells. Interstitial fluid is constantly changing or exchanging contents with the plasma. The interstitial fluid is also constantly exchanging components with lymph fluid. And then we have this other fluid compartment called transcellular fluid, which consists of cerebrospinal fluid bathing the brain and spinal cord, serous fluid surrounding your pericardium, your parietal peritoneum, and your pleura of the lungs. Synovial fluid, which you'll find in the joints, aqueous and vitreous humors, which you'll find in the eye. So all of these fluids are constantly changing, exchanging contents back and forth with each other. So whatever happens in one fluid compartment is going to have consequences in other fluid compartments as well. So be very aware of this constant exchange between fluid compartments. Now, primarily what's dissolved in water to make it fluid are electrolytes. And we divide them into extracellular electrolytes and intracellular electrolytes. Typically, what is high in concentration in extracellular fluids is low in concentration in intracellular fluids. And likewise, what's high in concentration in intracellular fluids is low in concentration in extracellular fluids. Now for this class, I'm not going to have you know all the specific concentrations of electrolytes in the intracellular extracellular fluid. However, I do want you to know which electrolytes are high in extracellular fluid and which electrolytes are high in intracellular fluid. And this is just a slide showing the exchange between interstitial fluid and plasma, transcellular fluid, remember that's joints, pleural membranes, things like that, intracellular fluid, and lymph. And notice that lymph exchange with intercellular fluid is a one-way path, and we'll talk about that when we get to that topic. All right, a brief discussion on cellular transport mechanisms. In other words, what are the mechanisms by which fluid and other dissolved substances get across the cell membrane so you can have exchange between the intracellular fluid and the extracellular fluid compartments. And these are divided into passive processes and active processes. So the first of the passive processes is diffusion, and in this case we're going to refer to this as simple diffusion. Why it's simple diffusion is because it does not need help from anything in order to, whatever is going across the membrane does not need any assistance to get across the membrane. It can go right through the membrane. And these are typically small molecules and non-polar molecules, or lipid-soluble molecules, because remember the lipid membrane of the cell is made of lipids, so lipid-soluble substances can go right through, and very small things such as oxygen and CO2, which we'll see in the next slide. Now, in order for things to diffuse across the membrane has to be permeable to that substance, and in the case of simple diffusion, the membrane is already permeable to certain things, and a concentration gradient has to exist. And what that means is that if there are 10 units of whatever this purple substance is over here, and there are 4 units of this purple substance on the other side of the membrane, then there is a greater concentration on one side than the other, and therefore, by definition of diffusion, particles are going to move from high concentration to low concentration. All right, so that difference in concentration is called the concentration gradient, and the particles are going to move down, in other words, from the higher number to the lower number, their concentration gradient. And as a reminder, this is going to happen until there's an equal amount of solute on both sides of the membrane, and we call that diffusional equilibrium. This slide is an example of simple diffusion across membranes using oxygen and carbon dioxide as example. And remember, those are two that can go right across the membrane. All they need is a concentration gradient because the membrane is already permeable to those substances. Now, last slide we talked about the concept of diffusional equilibrium. Equilibrium equals death for cells. So one of the ways that your body compensates for this is that you have things are always moving. So blood is always moving, and because blood is always moving, the blood flowing into tissue cells is going to bring a high concentration of oxygen. And since cells use oxygen, there's always going to be a low concentration inside cells. So there's always going to be this gradient between the blood and between cells that will allow oxygen to diffuse from the blood into the cells. And since cells are constantly using oxygen to produce energy and carbon dioxide happens to be a byproduct, there's going to be a high concentration of carbon dioxide in the cells, but a low concentration of carbon dioxide in the blood that's returning back to the lungs. So you're always going to have this concentration gradient that's going to pull carbon dioxide from the tissues into the blood. Now, our bodies tend to keep optimal conditions to keep diffusion rates relatively high. And three of those components that are part of this system are distance. In other words, low distance keeps diffusion rates very rapid. High concentration gradients keep diffusion rates very rapid. And relatively high temperatures keep the diffusion rates very rapid. The next type of diffusion is facilitated diffusion, and all the rules of diffusion for facilitated diffusion are the same as for simple diffusion. You need a concentration gradient and you need the membrane to be permeable to the substance. Now for those substances that are not soluble in lipids, in other words, those are too big or are not lipid soluble, they need carriers or channels to be able to help the charged substances or large substances get through. And so that's what we mean by facilitated diffusion. These channels or these carriers help the substance get through the cell membrane or diffuse through the cell membrane down its concentration gradient. Examples of this include all your major ion channels, sodium channels, calcium channels, potassium channels, and so on. Now, another type of diffusion is the term osmosis, and this tends to confuse a lot of people, so hopefully you guys have a pretty good grasp on this already. Osmosis is the diffusion of only water molecules, all right, only water molecules, but we're still going to talk about solutes because water is going to be attracted to solutes. So if you have a system with a semi-permeable membrane in there and you have 10 units of solute on one side and 1 unit of solute on the other side, regular diffusion says those solutes are going to move from high to low. But when we're talking about osmosis, we're talking about the movement of only water. And osmosis rules say that diffusion of water is going to happen from low solute concentration to high solute concentration. And the force that the solutes exert on the water to pull them towards one area is called osmotic pressure. All right, so this osmotic pressure pulls water towards a certain area. And it turns out that the higher the solute concentration, the more water is going to be pulled towards that particular area. And again, just like diffusion, this occurs until osmotic equilibrium is reached. In other words, there's the same solute concentration on one side of the cell membrane as there is on the other side of the cell membrane. But again, as we discussed, equilibrium equals death, even when we're talking about water. So there's always a tendency of water to move into and out of cells according to the solute concentration that's on either side of the cell membrane. Now, when water moves towards a high solute concentration, we refer to that high solute concentration as having a tonicity. In other words, the concentration of the solute is the tonicity of that solute. And the way we use that term tonicity is in reference terms of the fluid that's inside of cells versus the fluid that's outside of cells. So the first term, isotonic, means there's the same concentration of solutes outside the cell as there is inside the cell. And because there's the same concentration of solutes, there is not going to be any net movement of water across the cell membrane. In a hypertonic solution, we can't do anything about what's inside the cell. What we can change is what's outside the cell. And this is going to really come into play when you guys start learning more about IV fluids and whether they're hypertonic or hypotonic. And you're always talking about relative to what's inside the body or inside the cells. So when you put cells inside a hypertonic solution, the solution is going to have a much higher salt concentration than what's inside the cells, and water is going to move out of the cells towards that solution. And as a result, the cells are going to shrink or shrivel up or become dehydrated. In hypotonic solutions, the solution that is outside of the cells has a lower concentration of solutes than what's inside the cells. And if that is the case, then water is going to want to flow from low concentration outside the cells to high concentration inside the cells, which will cause the cells to swell, and if gone on long enough, can cause the cells to burst. So the last transport mechanism that does not require the use of energy is filtration. And filtration is basically molecules being forced through some kind of filter by some kind of pressure and separated by size. When we're talking about filtration in a body, we're talking about hydrostatic pressure pushing fluid that's going to separate what's dissolved in the blood across a semi-permeable membrane. So the example of that occurs in the body is when you have blood pressure in your arteries getting down to the capillary level, which are very leaky. They have pores inside of them, which we'll talk about in the cardiovascular section. That blood pressure forces fluid out of these pores. And with that, anything small enough to go through those pores is going to go with the fluid. All right, so you've just filtered out water and things that are dissolved in it that are small. What's left behind are cells and very large proteins, and that is by definition filtration. You filter out some things and some things are left behind. Now, an important thing to remember when we're talking about filtration in a blood capillary is that because proteins are left behind, they will then start to exert an osmotic pressure that will pull some fluid back in to the capillary on the venous end. And that's going to be important for the next thing we're going to be talking about in the next slide, which is edema. So edema is the formation of excess tissue fluid, in other words, what's directly outside the cells, the interstitial fluid, through a number of mechanisms. Some of those mechanisms could be increased capillary hydrostatic pressure, which is usually due to an increased blood volume or an increase in blood pressure. All right, some examples of what could cause this would be heart failure, which would lead to an increase in blood volume in certain areas, forcing more fluid out, or pregnancy, where you have an excess fluid volume as well. Loss of plasma proteins. Remember from the last slide, if we don't have as many plasma proteins in the blood vessels, you're not going to be able to pull as much fluid back in, therefore it's going to stay out in the tissue compartment or the interstitial compartment. Examples of that: burns, malnutrition, or another example would be liver failure. Increased capillary permeability, in other words, we increase the size of those pores in the capillary. This would be under conditions of inflammation, for example, and you got much more tissue fluid coming out than is what is able to be handled. Or prolonged standing or sitting, where blood tends to pool in an area and can't overcome the forces of gravity for one reason or another, meaning you're not really moving. So getting into transports that require energy, active transport's the most common one you'll hear of, and that's where there are these protein carriers, otherwise known as pumps, that use energy in the form of ATP to pump substances up their concentration gradient. In other words, if sodium is usually low inside the cell and high outside the cell, these pumps are going to work by active transport to pump sodium outside the cell against the concentration gradient. And the most common example you'll see is the sodium potassium ATPase, or sodium potassium pump, which pumps 3 sodium out against its concentration gradient and 2 potassium in against its concentration gradient. And this pump, which is present in all membranes in your body, is constantly maintaining that high sodium gradient across the membrane and high potassium gradient across the membrane. Remember again, equilibrium equals death, and your body uses a lot of energy to make sure that equilibrium is not reached so that cellular functions can go on. Now, another type of transport mechanism that uses energy is endocytosis. And endocytosis is a process by which the cell takes in particles in its environment that are too large to be moved by diffusion or by active transport. And it uses vesicles inside the cell to be able to do this. So what will happen during endocytosis is the cell will form an envelope around a substance, take it in, and then it will be inside a vesicle within the cell, and a number of things can happen from there, which we're not going to talk about, but be familiar with what endocytosis is. A type of endocytosis is penocytosis, otherwise known as cell drinking, and this is where the cell will take in fluid from its environment that contains things that are dissolved in that fluid into itself as a fluid-filled vesicle, and these could be ions, these could be a number of things that are taken in that the cell may need. Phagocytosis is by far the most common type of endocytosis you'll hear about. This is cell eating. Typically you'll hear about this with white blood cells, where they will take in solid substances from their surroundings, such as bacteria or viruses. And then once they are taken into the cell in the vesicle, they'll usually fuse with a lysosome. And remember, lysosomes have these digestive enzymes inside of them, which will digest that bacteria or virus, rendering it harmless, and will recycle the parts so that the cell itself can use them. And the third type is receptor-mediated endocytosis, which is a very specific type of endocytosis. In fact, you have receptors for specific substances on the surface of the cell, and once those substances are in abundance and bind to those receptors, the cell will endocytose and take them into the cell within a vesicle, and then deliver them to wherever they need to go within the cell. And some examples of this would be proteins, antibodies, hormones that interact with the cell and get taken inside by this mechanism. The opposite of endocytosis is exocytosis. In other words, using the same mechanism where you take a vesicle and spit the contents of the vesicle outside the cell. That's what exocytosis is. Think of it as the excretory system of the cell, or a way that a cell is able to release hormones or neurotransmitters through this process of exocytosis. Now, if you combine endocytosis and exocytosis, you get this concept of transcytosis. And transcytosis is using endocytosis to get a substance into the cell, move through the cell by vesicle transport, and then move out of the other side of the cell by exocytosis. And this is a way that certain things, for example, in the gut, can get across the gut barrier and into the bloodstream by using this transcytosis mechanism.